The A cell releases signaling molecules called A pheromones. A pheromones bind to A pheromone receptors on the alpha cell. Alpha cells release alpha pheromones. Alpha pheromones bind to alpha pheromone receptors on the A cell. The alpha cell releases signaling molecules called alpha pheromones. The A cell releases signaling molecules called A pheromones. Alpha pheromones bind to alpha pheromone receptors on the A cell. A pheromones bind to A pheromone receptors on the alpha cell. The ligands bind on the extracellular side of the receptor. The same process is triggered in both varying cells. When the signaling molecule binds to the pheromone receptor, the receptor changes shape. Inside the cytoplasm, the inactive heterotrimeric G protein, which has a GDP molecule attached, binds to the newly shaped receptor. This changes the G protein shape and causes an inorganic phosphate to bind, transforming the GDP into GTP, which activates the G protein. The activated G protein diffuses along the membrane and binds to the enzyme CDC42, changing its shape. An inorganic phosphate is then cleaved from the GTP of the G protein, making it back into GDP. The change in shape of the CDC42 enzyme causes the first inactive kinase, STE20, to bind and alters the shape of STE20, activating it. STE20 now begins a phosphorylation cascade. Activated STE20 binds to the second inactive kinase, STE11. An ATP molecule from the cytoplasm binds to STE20, which cleaves a phosphate group from the ATP, making it ADP. STE binds the inorganic phosphate of the STE11, triggering a shape change in STE11, making it an active kinase and causing the ADP to separate from the STE20 and is released into the cytoplasm. The newly activated STE11 binds to the third inactive kinase, STE7. Another ATP molecule from the cytoplasm binds to STE11, which cleaves a phosphate group from the ATP, making it ADP. STE11 binds the inorganic phosphate to the STE7, triggering a shape change in STE7, making it an active kinase. Activated STE7 binds to the fourth inactive kinase, FUS3. A third ATP molecule from the cytoplasm binds to STE7, which cleaves a phosphate group from the ATP, making it ADP. STE7 binds the inorganic phosphate to the FUS3, triggering a shape change in FUS3, making it an active kinase. The activated FUS3 kinase binds to the inactive transcription factor formin. An ATP molecule from the cytoplasm binds to FUS3, which cleaves a phosphate group from the ATP, making it ADP. FUS3 binds the inorganic phosphate to the formin, triggering a shape change in formin, activating it. The formin then diffuses into the nucleus.
The activated formin diffuses into the nucleus and binds to a specific segment of DNA, which causes the ACTB gene to be activated and eventually to be expressed as an actin protein. The ACTB gene is transcribed as a strand of mRNA, which diffuses out of the nucleus to the ribosomes, where the actin protein can be synthesized. The actin protein, synthesized in the ribosomes, causes a projection of the yeast cell to extend toward the opposite variation of the yeast cell. The SPA2 protein signals for the place where the actin should extend from. The protein CDC10 strengthens the structure of the projection. Actin extends, creating a shmoo projection from the membrane. The shmoo extends towards its mating partner. The mating partner is determined by where the ligand received by the opposite yeast cell originally came from. These projections from each yeast cell allows the cells to merge together and create an A-alpha yeast cell. The enzyme protein phosphatase dephosphorylates all of the activated proteins in the phosphorylation cascade by binding and removing a phosphate group from each protein. This causes the signal transduction to stop. The removed inorganic phosphate groups are released into the cytoplasm and are able to be reused and bound to ATP. The protein kinases are able to be reused after being dephosphorylated if they are phosphorylated again.